Once again, on behalf of APSE Organizing Committee, I welcome you all for this webinar on targeted nano-sized drug delivery systems and their therapeutic potentials. So I also thank the organizing committee for giving me this opportunity. So with this, I conclude. Have a wonderful day and thank you all.
Thank you, Dr. Pavan. It's a nice introduction about welcome note. Next, I would like, let me move with the session. Before going to move with the session, I request all the participants, and I would like to share some legislations over here to begin the session. Please mute your cell phones as well as your audio. And very important thing is, do not hesitate to question whatever it's rise in your mind. Let it be a very simple or a difficult. So only thing is, we encourage the questions have to be raised from each and every participant. So the important thing is not to stop questioning. So your questions can be mentioned, or you can write your questions in the chat box. At the end of the session, our speaker will discuss about the at the end of the session. So without wasting our speaker's valuable time, I would like to request Dr. Banappa Ungar, Deputy Director of ICMR NITM Belagavi, to chair this session. Dr. Banappa, over to you.
Thank you, madam. First of all, I would like to thank.
And I request Dr. Banappa to introduce our speaker.
Yeah. First of all, I would like to thank Professor Dr. Biswajit Mukherjee sir for accepting our invitation and to deliver this lecture and to be with us today. Thank you, and it gives me an immense pleasure to introduce Dr. Biswajit Mukherjee sir to the audience. Dr. Biswajit Mukherjee is a distinguished professor, accomplished scientist and a teacher of our pharmacy profession. He is a professor in the Department of Pharmaceutical Technology, Jadavpur University, Kolkata. He is a former DAD, that is a German Academic Exchange Service Fellow, Germany, and ex-guest scientist, German Career Research Center, Heidelberg, Germany. Additionally, he is a coordinator of AICTE sponsored quality improvement program, that is the QIP nodal cell in Department of Pharmaceutical Technology, Jadavpur University, Kolkata. And he is a joint coordinator of Center for Advanced Research in Pharmaceutical Technology, Jadavpur University. In addition, he is a convener, Animal Ethics Committee, Jadavpur University, and is the coordinator, Department of Science and Technology, FIRST program at the Department of this Pharmaceutical Technology in Jadavpur University. He is a former visiting fellow of the School of Pharmacy, University of London, London, UK, and is a former Indo-Hungarian Education Exchange Fellow, National Research Institute for Radiology and Radiohygiene in Hungary. He is also a recipient of Finland Government Scholarship, and he has worked as a guest scientist in German Cancer Research Center, Heidelberg, after receiving his Overseas Research Associateship Award from the Department of Biotechnology, the Government of India. He is also a former faculty of University Institute of Pharmaceutical Sciences, that is UIPS in Punjab University, Chandigarh. He also worked as a junior executive in the production in a UP drugs and pharmaceutical company. So overall, his career is the contribution to our education, science, it's voluminous. So in addition that it goes on, he works on his research contribution on antisense technology and chemo prevention in a cancer model. He has been working on novel drug deliveries, particularly transdermal patches and nano-sized liposomal and neosomal formulations, as well as nanoparticles. That is the topic of today's deliberation. He has supervised many projects, research projects funded by different funding agencies like UCC, CSIR, ICMR, DST, DBT, DRDO. So it's the number goes on like this. No, its contribution to the science, as I said, it is a voluminous. I'll be just listing out some of them here. He has guided number of PhD scholars, master degree students also. He has published his work in many international journals of repute. He has also written several books and book chapters on these different topics. He has a few patents in a process technology. He is an editorial board of some of these international journals of repute, and he is a recipient of UGC award, research award in 2009. He has received some other awards also. I'll be listing here some. Okay. So in addition, he has served as an editorial board member and editor, guest editor for many international journals. He is a section editor of Encyclopedia of Metalloproteinases. It's published in 2013 by the Springer. He has become a co-chairman of Scientific Service Committee in this local organizing committee at the 56th Indian Pharmaceutical Congress held in Kolkata in 2004. He is an honorary secretary of Indian Association of Pharmaceutical Scientists and Technologists, Kolkata. He has participated in different seminars, conferences to deliver invited lectures both in India and abroad. He delivered a prestigious invited lecture in the Presidential Symposium of a 62nd Indian Pharmaceutical Congress at Manipal in Karnataka. So the CV, now it goes on voluminous, as I said. I'll be just completing with a brief note on this. Thank you very much, sir, for accepting our invitation on behalf of this society. Really, it is a overwhelming to see your achievements, and it is an inspiration for all our scientists and teachers in the pharmaceutical field, sir. Thank you very much, sir. Welcome you for this lecture.
Thank you, sir. Thank you, Dr. Banappa, sir. And really, it's, as you mentioned, it's really overwhelming to have Professor Biswajit Mukherjee, sir, today on this forum. Because if you see his citations in the Scholar, it's more than by this year, it's crossing 3,500 citations. I welcome Dr. Joshi, Secretary of the APSE, for this session also. So without wasting our speaker's time, let me give it to the forum to our sir. Professor Biswajit Mukherjee, sir, you can share your slide now. Sir, can you? It's over to you, sir.
Thank you. Thank you. So should I go for the presentation?
Yes, sir. Yes, sir.
Just a minute.
Sir, you unmute your audio, sir.
Yeah, because I had to
Yes, sir.
go to switch on my power of the computer. I found that battery become very low, and I forgot to switch on the power.
Okay,
so
sir.
I left and I just did it, and then I actually first called my daughter, but she didn't turn up, and I went there, I switched it on, and then I came back.
Thank you, sir. No problem, sir.
Yeah. So I hope you will permit that thing. Yeah, people are joining here, Deepak Patil and all these things. So you please, you control them so that this should not come to my screen.
Yeah, I have kept this legislation in the beginning to the participants. Those who are here, please you mute your audio so that the speaker can deliver. We can hear this our speaker's lecture comfortably.
Okay, I can admit Deepak, but please don't allow the people to come just.
Yes, sir.
regularly, because that will disturb
Yes, yes.
the
Yes, yes.
presentation. Then important issue is that, first of all, I like to convey my sincere gratitude and thank to the President, Professor Milan Parle, and his entire team of, the Secretary and Joint Secretary and others. So yes, Dr. Mansadipa was instrumental to organize this, along with the Secretary also, and Dr. Manappa and Dr. Kumar, Pawan Kumar. I think they have completed my speech because they have delivered exaggerated introduction as well as the most important parts of the nanomaterial. So even if I don't say anything, possibly people have understood the importance of it. So thank you very much. And yes, Dr. Manappa, am I right? Manappa?
Yes,
Yes,
yes.
sir. Yes, sir. Yes, sir. Your
Yeah.
the biodata is itself will be an example that that what is the importance of this the topic it is, and you being with us definitely will be enlightening for all our colleagues on this platform. We will be enriched with the knowledge, sir, and we will get benefited definitely, sir.
Thank you very much. But the delegates who are there, for them, the information is, I am a man like you. Those who are sitting in the opposite side, the same role is mine. Sometimes I sit there or sometimes I sit here. What for I have come here? I have come here for just to share some of my thinking and thoughts and my findings with you. So once again, I like to convey my thanks. I have seen Professor Sarad Kade, yeah, from Dipsa, and it's nice to see because after a long time I saw him in the
eagerly waiting to see you and listen to you. Thank you, sir.
Yeah, nice to see you. So let me just start, because already we have spent some valuable time. You understand that my speech is quite interesting in the line of the topic. We have targeted nanosized drug delivery. And then their therapeutic potential. So two important areas I want to touch. One is the targeted delivery. Of course, this is nanosized. And the other is the, why my slide is not getting changed. Yes? Any problem? Have I shared my slide?
No, sir. We can see your slide, but you just click the next. Yes, sir. Yeah.
Now it has come.
Yes. Okay, sir.
It is okay.
So, yeah. So the areas I want to touch is, first of all, target, just to bring it to the right location. Secondly, the carrier, nano carrier. The carrier means which carries the thing, vehicle, which carries the thing from one part to the other. And third thing is, of course, this is related to drug targeting, that is to reaching the drug to the right area of the cells. This is very important. Now, why targeting? It is, I think all of you are well aware that we want to just send the right amount to the right location. I want to send something to New York, and from maybe from Calcutta, or maybe from Mumbai or Delhi, and the flight when reaches at New York, maybe 50%, 60%, 70% of my goods are lost. So only 40% is reaching to the target. And this, 60% is lost on the way. This is not wanted. This is not a good carrier system. Carrier system means if I send 1 ton of material, it will reach to the right direction and right place with this 1 ton only. So this is called the good carrier. So good carrier means it will bring the right amount to the right location. Unfortunately, this is a very, very difficult job. To bring the things to the right target in the body is always difficult. It is possible on the roadways, on the airways, on the sea, but it is difficult when you are considering our body. Why it is difficult? Because our body is very, very complicated. So many things you have to come across. For that, what we do, what normally we follow, you understand that we take Huge amount of drug, huge amount of drug, additional amount of drug. And you know that huge amount of drug means what? Say paracetamol, 500 milligram of drug normally we used to take. Nowadays, all on a sudden, I do not know what makes it, how has it been done, 650 milligram, 150 milligrams you are taking additionally. Let us consider your body weight. You are an adult, your body weight, based on your body weight, we consider the dose? No. You are an adult, and based on that, we consider the dose. So maybe your body weight differs, your height differs, but we are taking, all of us are taking 650 milligram of drug, paracetamol, one single dose. Question is, do we need it? Answer is no. Because our enzymatic system, particularly, in liver, phase 1, phase 2 enzyme, we don't need lots of, we don't have the similar kind of enzyme capability. So maybe I need less, you need more. So companies cannot prepare medicine for you and for me in a differential way. So what do they do? Normally they prepare this medicine in an average standard. But considering this average standard, you see what happens in our system. You understand this Avogadro's law. You understand that 1 gram mole contains how many molecules? 6.023 into 10 to the power 23. If you write on the blackboard, you cannot read it. If you write it on the blackboard even, you cannot read it. So it's a huge, huge, huge amount. So just write the figure 6.023 into 10 to the power 23. So just write the zeros. You cannot read the number. Such a big number. And I'm sorry, I cannot allow other people to enter. So if somebody else is there, please do it. Now, question is, every case, even if you take 10 milligram of drug, do you understand how many additional molecules you are consuming? Drugs are not your friend. These additional molecules will never do good to your system. And this will be definitely harmful for your system, for any drug, So remember, additional consumption of any molecules, or I should say rather, the consumption of any additional molecules. Additional molecules means after the requirement, what you are taking. It will not be good for the health. It will be harmful, definitely. And if you consume it for a long-term basis, particularly like the drugs of, anti-cancer drugs like, anti-diabetics, and also antihypertensive, we consume it for a long, long time. And that those additional molecules definitely be harmful for this system. So it is important at this moment, by the first slide, I hope I can make you understand that to bring the right amount to the location is very important. And for that, what we need, we have to do it. And for that, drug targeting is important. Formulation is important, but it is more important to prepare the formulation which will go to the right location. Otherwise, to disperse it throughout the body means it will harm to the other cells also. So we need the carriers. We are not working with the drug. Drug is not our focus. Our focus is the carrier system. Means like you want to send something from one side of the river to the other side, you need a boat. So boat is the carrier, not the human being. We are not working with the human being, rather with the boat, the system, carrier. And that carrier means from one port to the other port it is going. It is not lost, or it is not going somewhere else. So our target is we want to send the carrier from one particular port to the other. That is the thing. So you see, that's why in the system, drug targeting is our purpose. I do not know what happened. Why it is not changing? Yeah, so I have to click on this. So you see, yeah, so this is related to another important issue is the nano size. Nano size is nothing unknown to you. It's a very, very small size. And I have given you some pedantic information. You will get it from the internet. The size, the atom size is 0.1 nanometer. Carbon nanotube, it is 1 to 2 nanometer. Bacteria, normally it is 1 to 10 micrometer. Viruses, normally 40 to 100 nanometers. So you understand the advantage of virus than bacteria, because such a small size can penetrate into the cells easily and can also disperse easily than bacteria. So you see, bacteria chances of death is much more higher as compared to the virus, because their penetration power is much more because of the small size. And also the protein, 4 to 10 nanometer, which are controlling our body, every work, whatever they're doing, mostly, it is regulated by only one type of compounds, proteins. So we understand the importance of it. Important thing is then why we are studying the nanomaterials. Why? The important issue is nanomaterials has some advantages. When you are bringing the substance into nano size, surface area, this and that properties, it is nothing unknown to us. But the thing which is important, that that bringing nano size means you can get some extraordinary property which you will not get in normal cases. You can change the solubility. You can improve the solubility. Materials which was not supposed to enter into the cells, it will enter into the cells because of the nano size. Material which is not supposed to cross the blood-brain barrier, it will cross the blood-brain barrier due to nano size. So anyway, it is super magnetism sometimes is produced on the surface. So there are new properties we can get. These properties, whether sometimes it is helpful or sometimes may not be helpful. So by tests, by findings, by experiments, by investigation, only we can try to understand. They generate some surface charges because of the material bringing into nano size. So you see, I can give you one simple example, very interesting example. See, gold. Gold, they are not dissolved in water. They normally, in aqua regia, 3:1 ratio of hydrochloric acid, nitric acid combination. Their gold dissolves. But you see, if you bring it into nano size, and depending on the size, you can get the different colored gold solution. You see the different colors solution is there. You see red, black, white. So you can just dissolve the ring, gold ring, simply by bringing it into nano size without taking the help of aqua regia. So do take care. If somebody can convert your gold ring into nano size, it can be dissolved. So you see, this is the advantage. This is the thing which you can generate, you can produce, and that's the thing I believe it is very important. Now, the next important issue is when we are just considering our formulation, we are now tending it to formulation, because the thing whatever I have given you so far, this is related to the pedantic information, available information, and what I call bookish information. Now, what I want to tell you, the findings, what we are going to do. We have been working on this field with lots of different targeting materials, but here I want to focus maybe one or two cases because of the shortage of time. One such case, basically, we are applying Aptamar. What is Aptamar? Aptamar, you have to understand, they're small nucleotide sequences, small DNA sequence or RNA sequence. They're called Aptamar. Small sequence. Maybe small means even sometimes 20, 30 bases. And what is the advantage of the Aptamar? It's a nucleotide sequence, single stranded sequence, but they form certain kind of folding. Because of that folded structure, it's not a linear structure, folded structure. So because of the three folded structure, they can bind with certain proteins, different proteins. So it is very specific, like monoclonal antibody. So aptamers, that's why it is called, chemical or synthetic monoclonal antibody. Very interesting. The structure, when you are just preparing this small single-stranded nucleotide and they form a 3D structure. That 3D structure, sometimes they form the structure in such a way that can bind to the exactly as antibody binds to the antigen. So with a specific structure, they just fit to that particular area. So thereby, aptamer can be very specific, very targeted to a particular protein. So aptamer, I said, single-stranded nucleotide, DNA or RNA. Now question is, single-stranded nucleotides, if you inject it into the body, it will be degraded because there are enzymes like DNAs, RNAs. So these enzymes, single-stranded nucleotide will be split. They will be destroyed. Then you have to protect it. How can you protect it? Can you change the bases? Answer is no. You cannot change the bases. Because if you change the base you understand, adenine, thymine, cytosine, guanine, their base. So uracil. So you see, if you change the base, there will be a perfect mismatch. There will be a mismatch. So it will not fit to the right target. So you cannot change the base. Can you change the sugar moiety? No, because deoxy sugar is specific to DNA, ribose sugar is specific to RNA, and because of this sugar also, they can give you this 3D structure. Then where you can change? You can only change in the phosphodiester linkage. That is, I have shown you here, you see, here is phosphodiester linkage. So one of the oxygen you can replace, or sometimes this is replaced. What happened, these DNAs, they cannot recognize this because they don't have eyes. They only recognize this sequence and split it. So, they cannot recognize this phosphodiester linkage because you are modifying. What we do, we just take the help of the modification of this, one of the oxygen, which is replaced by sulfur, It is called phosphorothioate. Yes. So it is called phosphorothioate. So this phosphorothioate linkage actually, the linkage we can modify, and thereby you can make the aptamer stable, this single-stranded nucleotide stable. So these aptamers, you see, when you are just attaching to the nanoparticle surface, they can act as a targeting molecules, and inside that, you can just put the drug, what I have shown you in that figure. So many things are coming. I do not know you can see my slides or not. Yeah, so you see, Jitendra Kumar Gupta, these people are joining and joining and joining. So you see, this is the work, what I am going to represent. This is already published, molecular therapy and nucleic acid research. We have published this very recently, last year, 2020, middle, June. So you see, nearly 1 year before. And you see, this work is I am going to present today. This is just aptamer mediated targeting of nanomaterials to hepatocellular carcinoma. Why I am just sharing this? I'm sharing this because you see, during my lecture, whatever I will say, after that, if you have any question, or if you want to know the details of the study, or the, what you call these methods and all these things, you can consult this, you will get the detail of it. It is really available online. Now again, the problem is I have to click or what I have to do. Yeah. So just see the formulation. The formulation, nanosized formulation, whatever we have prepared, there's scanning electron microscopic view at the left side. Then you see the next one is STEM to understand what is there actually from inside, and in the AFM just to see the surface and distribution. So this is the normal study. We have to do it to understand the size of the particle, etc., along with this other common studies like particle size analysis, etc. So the finding is important to understand whether they are really nano size or not. But the next important issue is whether the aptamer is conjugated or conjugated to the surface of the nanoparticles or not. That is more important. We have used actually several aptamers, 5 different aptamers we used. Out of these 2 different aptamers seems to be quite satisfactory. So we are representing these 2 aptamers. One is L5 and the other is L2. And remaining were not very satisfactory. So you see, along with that, we also studied with certain proteins like galactosamine and transferrin. So we used also. So it's a very comparative study to see which one could be the most beneficial to target. So here you see the well is illuminating. This wells contains the nanoparticles containing aptamer. And these are free aptamers. 2 different aptamers we have used. So wells containing nanoparticles, they could not move because of the heavy weight of the nanoparticle, but the aptamer freely moved. This is the free aptamer. This is the conjugated nanoparticle, aptamer conjugated nanoparticle. These are the free aptamer. And these are, the ladder to understand this size of the aptamer, whatever we have used is correct or not, etc. This data suggests that yes, it is conjugated perfectly. Then we have gone for the investigation. What investigation? In case of a study, you have to go for first in vitro analysis, then you should go for in vivo analysis. Based on the success of the in vitro analysis, we go for in vivo analysis. This is absolutely, you understand, the in vitro analysis. In the in vitro analysis, what we did, you see, basically, it's a quantitative uptake. First of all, we want the cell is taking up or cell is at all internalizing the aptamer conjugated nanoparticles. So I told you, we have used several different kinds of aptamer, 5 different aptamers, and then free nanoparticles. See, you see, this is PTXNP. This is 3 nanoparticle without any conjugation. Then this is your L5 conjugation, L2 conjugation, L1 conjugation, L4 conjugation. So we have used the different conjugation, and we have got the uptake, cellular uptake by FACS, flow cytometer. You understand? It's a very sophisticated instrument. 2.5 crore is the cost. So you see, the findings suggest, you see, L5, it has a very high uptake, followed by L2. Remaining is L1 is there, but you see L1 is already, having almost similar findings with L2. So we discarded L1 later on. So you see, these 2 cells, A and B, basically we have taken the HepG2 cell, and HUH cell is, HUH7 cell is the C and D. Basically, all these cases, you see, the uptake is very high, particularly in case of L5, followed by L2. So L5 was very satisfactory in case of this HepG2 and HUA cell. HepG2 and HUA cell, what is that? Both of them are, HCC cells, cancerous cells. If you want to, then we have gone for this, what you call this IC50 value. uptake, and then IC50 value. And in IC50 value, that is, we have studied in this is a PF is marketed formulation, and then NP, this is normal formulation, and these are the different formulation, and this is protein related formulation. So that is, I said that one is galactosamine and the other is transferrin. So we found that only this particular L5 and L2, these two combination is having, the potential to kill the 50% of this 50% of these cells at a very, very high degree. Interesting thing is that you just see, we have studied it in different cells. HG2, HUA. So HG2, H7, these two are the cancerous cells. Chang liver and WRL68, they're the normal cells, normal hepatocytes. So you see, we have compared Chang liver and the normal hepatocyte cells, as well as we have compared with the other cases. So we found that yes, we have selected these two as the best formulation. Now we have to go to the next, but I do not know what happened. Yeah. So here you see, then we've have gone for the quantitative uptake in this Chang liver and WRL68. So quantitative uptake we have seen earlier in HUH, and that is HEPG2, this cancerous cells, and here it is normal cells. You see, very, very interesting, very, very interesting. Quantitative uptake in L5 is, and also L2 is more or less same as control. Quantitative uptake is almost not there. That means there is something in cancer cells which actually insisted, or rather initiates, or rather induced, or rather mediated the uptake of this L5 and L2 conjugated nanoformulation in the cancerous cell, which is not happening in the case of the normal cell. So normal cell uptake is similar like controls. The uptake is very, very similar like control uptake. This actually encourages us that we should select this formulation, and this one, because they were very effective, we found, in case of the cancerous cells, but not, creating problem for the normal cells. So targeting purpose, this is very good. So we have selected this particular two formulation. Now you see, then another important issue is that here you see, we wanted to see the what you can say, the cellular or cell cycles. We wanted to see how cell cycle is behaving with the formulation. And here we have also taken all the formulation, including this protein conjugated formulation. They were not found to be very effective as compared to Aptamar. So, so what happened, if you just follow the data, you can see that control is here, and L5 conjugated is here. These are, the commercial formulation, free drug, and this other formulation, this is there. In case of this A and B, two different cancerous cells. A type is, this HEPG2, and this HUH7 is here in B. Both the cases, you see, the arrest or the cell cycle arrest is mostly in G2M phase. You know that there are 4 phases, actually 5 phases, but 4 phases normally in the cell cycle. This 4 phases is what? Most important phases is G1, S, G2M. And G1 is the after cell division, the normal growth phase. S is the before cell division, the DNA synthesis occurs. Then cell prepares for the mitotic division is G2. And finally, you can say that M phase is mitotic phase. So you see G2M phase, huge amount is 90, I think 55% here. In this case also L5, this G2M phase is 32% here, S phase is 42. So you see basically, G2M phase in this particular case, more I like to say about HepG2 cells is mitotic process is being arrested much more. Of course, it is being arrested also in case of these HUA cells. So maybe little at a slower degree, but definitely it is happening because this is also a very high value. So you see, with the time, what we understand about all other formulation, you see, L5 has much more, G2M phase arrest, and that indicates, this paclitaxel drug we used in the formulation, and paclitaxel, it's a known drug for arresting the metastatic, I beg your pardon, in the mitotic phase, mitotic spindle division. So here you see, this is this proves the drug is working rightly in those type of cells, cancerous cells. Yes. And then this is once again, yeah. So this is once again we have studied for particularly the DNA fragmentation assay by, Viadu DNA fragmentation assay. Sometimes you can say tunnel assay also. So you see what our finding is. Once again, if we go for this L5, you see DNA fragmentation is 86,000, and it is around, it is around similarly 86,000 something, 800 something. But in other cases, it is just 4,000, 4,700. So, but if you go for this L2 is 51,000, 57,000 in these two type of cells, A and B. A is FG2, B is the HUA. So you see, what I am telling you, the huge amount of cells, actually huge amount of DNA fragmentation is occurring with these two formulations compared to the other. That indicates, the cells has lots of DNA fragmentation, means the death process of the cancer cell is there. These people are, why they come and join at this time, I do not know. So you see, the study related to apoptosis. The study related to apoptosis is what? We so far found some important issue. What are those issues? One issue is the cancer cells, these particular formulations are going much more inside the cells. Internalization is much more as compared to normal cells. Second is what? Secondly, the DNA fragmentation is much more in case of the cancer cells than the normal cells. So obviously we wanted to see whether the apoptotic cells, apoptosis is occurring or not. Why this is needed? Why we are going to study apoptosis? The reason is that if we found the cell is dying because of the necrosis, your formulation is not at all good, it is better to stop, or you better to modify, because necrosis is a very, toxic death, and this is never wanted. That you should kill the cell in an apoptotic process. So in the normal programmed way, you have to kill the cells. This is how we can make the body, healthy. So if you want to kill the cells, just putting some nitric acid, you can kill the cancer cell. But this is not possible. That type of death is necrotic death. Or any toxic chemicals, if you add on to the cell, it will die. But this is necrotic cell. It can be very harmful for the other normal cells also. So it is never wanted. You have to kill the cells by apoptotic means. Apoptosis is very important. And for that, what we did, we studied the apoptosis But before discussing this, I want to tell you important issues. This is Hep G2 case. And you see, we have used the different formulation, and this different formulation, even out of this different formulation, this is the normal cases, our formulation of interest is, L5 and L2, these two. What happens, in normal cases, how do we do it? This acridine orange and ethidium bromide staining is done. Acridine orange, ethidium bromide staining. In normal cases, it is the green appearance is there. In case of early apoptotic, it is also green. Late apoptotic, you will get orange to red. Now you see, and the necrotic is also red. So in terms of color, I just want to tell you again that when you are observing in terms of color with the acridine orange and ethidium bromide staining, and these are all confocal microscopy. So you see the cells in normal case, you see, you will get the green. Apoptotic case, you will get the green. Early apoptotic. Late apoptotic, it will be orange to red, and necrotic is red. Then how can you differentiate? You see, in normal case, it is green. Apoptotic, you will get bright green, but there are with some characteristics. One is, the chromatid condensation. Chromatin condensation is there. Membrane blabbing, membrane disintegration, cell shrinkage, or you can say other like formation of the apoptotic bodies, and so nuclear fragmentation, this is there. So, and then you see late apoptosis, yes, it will be red, but orange to red, but of course, it contains this chromatic condensation and DNA breaks. So obviously this happens, and also the sometimes shrinkage of the cell. So the character of the earlier, what I have described for the early apoptosis, late apoptotic, also similar findings is there. But necrosis is red or orange, but the nuclear morphology is same like normal. Nuclear morphology is cell, you will get like normal. So you see here lots of condensation, quantic condensations, and the fragmentation, formation of apoptotic bodies. So if you will find, so this is there. And in terms of this late apoptosis, particularly L5 and L2 formulations, they have much higher degree of you can say late apoptotic phenomenon. So this is we have investigated in Hep G2 cells. Now if we go for the other, that is Hua cell, you see this is also we have similar findings in Hua cells. This L5 and L2, lots of late apoptotic cells are there as compared to the normal PTX nanoparticles, and also L1 and marketed formulation. So these are different findings we have achieved, but these findings what in both the cancerous cells shows that yes, the cancerous cells has, or you can say all the cancerous cells, particularly Hep G2 and Hua cells, they have, when treated with this L5 and L2 type formulation, they have much more late apoptotic cells. If I combine them, these two, whatever you have seen in Hep G2 and Hua, this is the picture. Overall picture, what I have shown you earlier, this in a single slide, it looks like that with the different formulation, where you see we have given even this what you call this galactosamine conjugation, and also the other conjugation like transferrin conjugation. All the data here you can see given, and here also you can see this L5 and L2, they have remarkable changes compared to any other slides. Then what we did, we wanted to see the changes of the mitochondrial membrane potential. Mitochondrial membrane potential, how we do it with this annexin FITC PI staining, and this is evaluated through this FACS, that is flow cytometer. And here also, you see, we have different types of formulations, and we have studied at the different phases. Here you see what happens in case of this mitochondrial membrane potential. We use this Gc1 staining. Gc1 staining, what happens? Gc1 staining is basically, it's a monomer. Gc1 is a monomer, and Gc1, when it enters into the cells, enters into the mitochondria, if it is membrane potential is lost, membrane disintegration is there, or rather membrane integrity is lost. In that case, the GI Jc monomer, Jc1 monomer, that becomes Jc1 cluster, or conglomeration is there, which forms the red color. So with red to green ratio, if it is more, that indicates the loss of mitochondrial membrane potential is more. So we have investigated this study, that mitochondrial membrane potential. And then you see, these people are actually blocking my slides. Who is he? Let him join or let him go. Yeah. So you see here, this is L5. Here also L5. So here finding is, you see, red to green, much more red. That means, L5, L2, they have, you will get the value also with this mitochondrial membrane potential. And the upper part here, actually we have studied with the P3 and P4 compartment. P3 compartment shows your live cells, and P4 actually apoptotic cells. So P3, P4 ratio you will get in the other case. So thereby we can understand that once again mitochondrial membrane potential and apoptotic process, which supports that the formulation like this L5 and L4 is much more potential in terms of the what you call this loss of membrane potential, and that means, and also induction of this apoptotic process. Now, if we go for this ROS analysis, ROS is very important. There's in the system, whatever happening, most of the cases, particularly related to disease, ROS has enormous role. ROS is reactive oxygen species. So we have investigated it once again, and this is also through FACS, and you can get the value of ROS, changes in the value of ROS, but I cannot show my slide because the upper portion is always blocked by entering people. So this is L5, it is 41,000 in case of this HUH cell, and here, who is here? So let them. So you see, this is 44,000 and 41,000 compared to others. Others is 7,000, 6,000, 15,000 maximum here. But here, you see, the values of different formulation is there, but it is 41,000. It's quite high, ROS generation to lead the apoptotic process in the cancer cell. So this high ROS generation, it is also there, I think 38,000. So ROS generation is very high in terms of these two formulations. These are the picture of the normal cells. Normal cells, that is the Chang liver, and B is the WRL68 normal cells. And here interesting result is there, that you see when you are treating it, the control cells and L5, no change. No change. Control cell L5, no change. Similarly, control cell and nanoparticle with drug without the Aptamar conjugation, no change. And then you see, this is also Aptamar conjugate, nanoparticle with the Aptamar conjugation, without aptamer conjugation, no change. So in the normal case, this is with the aptamer conjugation. But the commercial formulation, marketed formulation, you see they're causing apoptosis in the normal cell also. So this is dangerous. You see, they're killing the normal cells. That's why here once again this proves that yes, you need targeted formulation. Otherwise, you see, this slide shows that the damages are there in your cells. So if you take a cancer commercial formulation, whatever available nowadays, and if you apply it, this will damage your normal cells. This is very, very, what you call is very, very bad for the cancerous cells and normal cells also. I mean, bad for the cancerous people, because you are killing the normal cells. I'm very annoyed with this number of people are joining and getting lots of nuisance in my slide. So you see, when people are presenting, it should be free from all this, unwanted thing. That should be taken care of. Now you see,
sir, sorry to interrupt you, sir. We'll take care of that. Their entry, you need to mind their entry, sir. We are taking care of their entry, sir.
It should be taken care of at the beginning. At the beginning, yes, at the middle of the presentation, continuously, if people are joining, yes, the person who is presenting it, concentration of mind is distracted 100%. Because every time I want to present something, my slides are being blocked by some such thing. So this is not wanted, and I am really still they're coming. Either you block them, don't allow them to join. There's no point of joining at the middle of the lecture. So I do not know. Or if you want to allow them to join, let them join by other means.
Yes, sir. Yes.
Every time this is coming. So anyway,
sorry for the interruption, sir.
Yeah. Okay, forget about that. So what I was telling you, basically, in the normal case, we found that the formulations, conjugated formulations, and behaving like very, very normal way, that is entering into the cells, is not that good. But in case of the formulation, commercial formulation, they are being taken up not only by cancer cells, also by normal cells, and lots of necrosis and lots of apoptosis is there in those cases. Now, I will go to the next slide. Here, whatever we have discussed, and this is mostly we have mentioned here, and particularly we found in a single word, I can say L5 and L2 having potential, and this proves by this in vitro study. Let us go for in vivo study. If you go for in vivo study, and if you want to see what is happening, we have developed this cancerous model with this chemical carcinogen, 2-AAF, 2-acetylfluorine, and this is a very known, complete carcinogen. And also we have first induced the cancerous process with DENA, diethyl nitrosamine injection, intraperitoneal, single dose, 200 milligram per kg. So once this is induced, after 2 to 3 weeks, we have started the chemical carcinogen. After 20 weeks, then there is a gap of 2 weeks, and then we started the formulation up to 36 weeks. And that not regularly, I think twice or thrice a week, it depends on different formulations. So we normally use it, and then after that, at the end of 38 weeks, we have, killed the animal. So, and then perform the experiment. What we have found, this is the, cells, this is the, these are the cancer control cells, and you will get different types of lesions, in the cancer cells. We say they're all hepatic altered foci. First one, you see the hematoxylinosin slide. You can see there are lots of ground glass appearances there. They're early lesion. They're called early lesion. In a cancerous process, whatever may be in the solid tumor, there is a very nice sequence. It started with the glycogen stored foci, glycogen storage foci, and then eventually glycogen is lost, and then they develop, the mixed cell foci, focal lesions, and then this focal lesion converts into, basophilic lesion. No glycogen is there. This becomes basophilic with the toluene in blue, and then you see ultimately HCC, hepatocellular carcinoma. Anyway, what I wanted to see, this is a ground glass appearance, ground glass lesion. It looks like that cells are void, but it's not void. Actually, they have lots of glycogen storage, which is not being stained by hematoxylin, and that's why it looks like that. And then you see this is a basophilic lesion, basophilic areas, and then there are other lesions also. This is, this is called, it's a kind of, it looks like void. Void means nothing is there, but there it is not that. They are all cells, much enlarged, ballooning of the cells, and these cells sometimes have even more than one nuclei, you can say, and they remain always at the periphery. So you will get small nuclei at the periphery, and they also store a huge amount of glycogen. So you see, these cells are called, Spongiosis Hepatitis. So this type of cells, actually they're the precursor cells, you can say. They're the precursor lesion, precursor cells of the cancerous process. So these cells, basically the Spongiosis Hepatitis, these cells basically, what happens, whenever you see this in the liver slide, it happens in human, it happens in animals also, you can be sure that this is, in your case, the cancerous liver cancer process has already been started, because they are called the precursor cells of HCC. So this is bad. But anyway, these are these different lesions what I have shown, because all these lesion types we counted, and we considered this lesion type, cancerous lesion type, as hepatic altered foci, hepatic altered foci. And this is actually in case of, we quantified those and we have represented in the tabular form. I will show you the table. Here also this is called the normal cells, and after the treatment, the changes in the cells. See, after the treatment, how the cells has changed a lot, particularly towards its normal direction. So that means, you see, if you really target cancerous cells with the cell 5, more particularly, then it will be very, very beneficial. This is I do not know whether you can see it or not, but whatever I have showed, I have told you a little earlier that we have quantified it, and we have given here this type size also. This is less than 1 millimeter to greater than 3 millimeter, and in between also. So, and their number and counting is there. They're all HAP is hepatic altered foci. All this lesion, altered focal lesion we have considered, and we have given in terms of percentage. And you see in terms of percentage, or in terms of how it is changing. When you are applying this L5, you see the early lesion is much, much more, and late lesion is much less. But whereas in other case, you see cancer and other case, you see late lesion is more. That indicates that if you really apply targeting system, it will definitely delay the cancer process. Then what we did, we studied different, apoptotic and anti-apoptotic proteins, and we have found out their ratios, and we wanted to represent it, particularly this apoptotic and anti-apoptotic protein. Because what we have established earlier, that yes, apoptosis is there in terms of this, what you call this staining of the cells. But here, by the molecular marker investigation, you can say, you can make it much more supportive as document, because yes, this is actually happening because of this type of apoptotic protein induction, or this type of anti-apoptotic protein reduction. So on the whole, we have presented it, and you know that what we have found that actually it is supporting that yes, L5 and L2 are working based on, I mean, L2 formulations working based on the, you know, apoptotic process. This is now we have gone for the what you call the pharmacokinetic investigation. Here, pharmacokinetic investigation, here also what we have studied, you see, in the all these cases, the in uptake of the cells, particularly uptake means what we mean to say this PTX, that is the formulation, and this uptake has been, you know, there, and with the nano formulation, you know, this is lingering. This is lingering as compared to the normal formulation, which is just getting ended up by even around 40 hours or before. So you see the different nano formulation we have also investigated here. Unfortunately, these people are still joining. So after this is another histopathology of normal case, normal you know, rats. This is particularly we wanted to show that after the application of this L5 formulation, it doesn't create any problem of the hepatic architecture. That is in the toxic problem, toxicity effect we wanted to investigate, because we are now in the formulation side. We have also investigated different enzymes, liver enzymes, to see that whether this formulation in normal case, whether creating any problem or not. And this control with this, all this study with L5 and L2, you see, it is exactly reflecting that it is not creating much problem as compared to the commercial formulation. Here you see in 28 days commercial formulation 294, whether it is 222 and 213, it is more or less very close to you know, the normal cells, whereas it is very high in case of this commercial formulation. So I want to mean that in the cases of particularly if you go for this alkaline phosphorus, similarly it is true in other cases also. See, it is 96 and it is 70. So it is 122 commercial, but in case of this L5, it is just 70. So that indicates that SGOT, SGPT, and alkaline phosphatase, this which are considered to be the marker of liver damage, they're sufficiently, you know, protected when you are just applying this formulation. Oh. And then L5 formulation, of course, we found out of these two, L2 and L5. L5 is much more potential in this hepatocarcinogenic process. Then what we wanted to know, fine, we have engaged this Aptamar for targeting the liver cancer cells. We have used several formulations, and we wanted to see what happened, where actually it is targeting. And for that, we use this homology model, docking model, and we wanted to see actually which protein or what are the proteins where our Aptamar can bind. Let me tell you, we have selected 5 different Aptamars and 2 different proteins for this investigation. One is galactosamine and the transferrin protein for targeting purpose, and 5 different Aptamars, L1 to L5. These all 5 different Aptamars have been reported in the literature, but this L5 formulation was not been investigated so far. So that's why we picked up that L5 to find out whether really it can work or not. And we found that yes, it is working. And L2 was that particular Aptamar which has been reported that it can work the best. So we compared with L2, means it is we compared with the best. But unfortunately, No literature is available where they have said that AL2 binds with such and such protein to target the cancer cells, or L5 binds with such and such proteins to target the cancer cells. They said that targeting with, is possible with L5, but nobody has investigated. And L2, largely investigated, they found it is working, but nobody found the target. Nobody wanted to see the target. And you see, then what we did, we have gone for this. And by homology model, actually, I took the help of my colleagues, and because of it, because I'm not expert in the field. So what we found, yes.
এইসব কি সব ফটো না, ওটা প্রোটিন সিনথেসিসের ফটো।
Please allow the speaker to speak. Then we please unmute. Please mute her audio. Yeah.
Some very pathetic music is coming. Probably somebody is very annoyed with this long lecture, and he is willing to listen to some pathos. Anyway, it's good. And somebody, yes. Anyway, what I was telling you, that if we just put in the homology model, and we found that yes, two different proteins. One is, HSP, that is heat shock protein, and the other is TAC72. Can you remove this one sets of things which is coming on my right side, because this is blocking my slides. Yes. I do not know. You should take care of. This is annoying me.
Yes, sir. Yes. I do not know why today. I think the participants, please, I request in the beginning itself I have given, please not be lecture. Sorry, please, sir.
Anyway, so we have actually investigated with L2 and L5, and we found that heat shock protein 70, HSP 70, and another is this TAC72. These two proteins are the possible target protein of this particular aptamer L2 and L5. We are working on that still, and we wanted to find out really they are binding with this. I will just say another investigation in a very short manner, so that nothing to worry, and I will stop. That part, whatever I have showed, that basically this is related to aptamer conjugation with this nano formulation, and this is bringing to the right location, and you have seen the data that it is definitely improving the cancerous process, particularly liver cancer process, HCC. Now, another thing, what happened?
You are audible. Slides are also visible. No problem.
Somebody said you make your audio visible. My audio is now visible?
Yes, sir, we can see your photo as well as the presentation.
Okay, so somebody wants to see my photo. This is very important. Now, you see, what I was telling you, that, I want to go back. Here, this study is a very interesting study. This is absolutely not related to the target. This is also targeting, but not using the targeting molecules. What is there? You see, here we have developed different types of formulation. This is also nano formulation, but not nanoparticles. Particles, you understand, it's a solid, it's like marble. This is nano vesicles. What you call is nanoliposome. Nano vesicles, basically, it is soft material like balloon. Inside is hollow, very soft. So there's a membrane. So nano vesicles. You see, we have prepared nano vesicles, particularly, when you will prepare this nanoliposomes, it is easy to prepare. Second thing is, they are very uniform than the particles. to prepare nanoparticles is much more difficult in the in uniform range. But in case of this nano vesicle, you can get it. You can see this slide, and you will see it also again in the next slide. So these are nano vesicles, and you remember this particular chemical, 1,2-disteryl glycerol, then phosphatidyl ethanolamine. This is DSPE. You remember this chemical. This is very important chemical in this study, DSPE. These are the formulation you can see, and I do not know, possibly in this small picture is not visible. The size is only 30 nanometer, 30 to 35 nanometers, I think. This is very, very small size. These are all nanovesicles, very uniform. And you see, when they are, dispersed in the liquid, the size is little bit enhanced, because that was SCM in dry state, powder state, and when you are putting it in the liquid, they will swell little bit. And there, really, in the second case particularly, you see the B case, second formulation, you see it is below 100 nanometer. It is the average size is around, say, 70 nanometer. And we have prepared different formulation. Now you see, then when you are going to prepare this nanoliposome, one important issue is that you have to see the integrity of the membrane. It's a very, very small size, very, very small size. 30, 40, 60, 70 nanometer is, you understand, very small size. So how you can see it? You can see it by means of some dog is howling. So take care. So you see, please mute your audio. So what happens, that when you can see this vesicle, you have to see the membrane should be intact. And how it should be done, you have to use the cryotem. Cryotem you have to use, and you see these pictures are all made of cryotem, and see the integrity of the membrane in every case. No case the membrane is lost. And also we have got some bilaminar formulation. Bilaminar formulation, normally what we studied, that after 1 micron and above we can get it, but this is not true. Even below 100 nanometer and 30, 40 nanometer range also, you can get this, but this percentage is very, very less, even less than 1%. So that's why probably people don't consider. What happened? We have used FITC with the lipid, and we wanted to see whether our formulation is reaching brain or not. We wanted to administer docetaxel across the blood-brain barrier. So we have just loaded a drug in the formulation, and then we have used FITC, and then we wanted to see it is reaching in the brain or not. And we found, yes, it is reaching in the brain. Then interesting part is this one. This slide, you can see 1, 2, 3, 4, and 1, 2, 3, 4. See, this is normal. This is just we have given the drug. These are all technetium level products. So we have given the drug, and drug is not reaching in the brain. Second case, you see, we have given the nanoliposome, and it is reaching in the brain. But the quantity is very less, very, very less. What we did, we used, that DSPE. DSPE is a lipid, and that lipid we have used along with this membrane phospholipid of the liposome. And that why we have selected that DSPE. We found that normal liposome is reaching in the brain, but the quantity is much less. So what we wanted to do, how we can improve the quantity in the brain. And we have just used DSPE, because DSPE is lipid present in brain. So just by simple thinking, that if we can use this brain lipid with the formulation, it can reach in the brain. And let us try, let us investigate. And we found, yes, this is reaching nearly 400 times more, formulation is reaching in the brain. So this is another way we can go for targeting. Always it is not that you have to target your formulation with the very specifically with the targeting molecules. You also, by other means, using the different chemicals, using the different methods, we can also target the formulation to the right direction. So here also you can see the data, plasma data and brain data. You see the plasma data and brain data. If you see, the formulation is actually in the brain, or when you are giving in terms of this DSP, you see it remains in the brain for much longer time than the normal formulation also. That indicates, you see, that because of the presence of DSP, it lingers also the stay of the formulation in brain. So more or less whatever I have discussed two targeting two formulation, which is targeting one case in the liver cancer cells and the other case in the brain. So you are delivering the drug across the blood-brain barrier at a high amount. So you see, the simple thing is that it is quite possible that we can do it by just simply using our thinking technology, whatever it is, and it will be, I think, useful, because in future days, what we want to deliver the drug to the right location. This is very important. I want to show you my university, my department. It is here, and the stamp published by Government of India of Jadavpur University in its 50th year. So more or less, I think I have completed my lecture. I believe that you may have lots of questions in your mind, so do not hesitate. So you may please ask me if you have any questions. Thank you very much.
Thank you so much, sir. It's a really informative talk. It's no doubt about that. This need not to express that it is an informative talk. Everyone might have understood that it's really an informative talk. Participants might have been overwhelmed by presentation. And the thing is, I really apologize for the interruption in your lecture. I request Dr. Banappa, sir, please chair the session for the question and answer session. Dr. Banappa, sir. Yeah. Please unmute your audio, sir. I think.
Thank you for allowing
Dr. Banappa, sir.
Yeah. Oh, sir. Thank you for this elaborated, very nice talk on this topic, sir. It's a very, as I said in the beginning, that nano-sized drug delivery formulations are the need of the hour also, I can say that. And you have rightly pointed out that anything excess is a poison, and reducing its size and taking it in an appropriate amount, in a reduced amount, is a need for the better therapeutic outcome. So for this delivering targeted nano-sized drug delivery systems and their therapeutic potential, sir. Thank you, sir. We have some questions, sir, from the audience. I'll read one by one, sir. So from the Gnanachandra, Dr. Gnanachandra has asked that, sir, please share your experience in developing nano-sized, 1 minute, sir. Developing this oral insulin, sir, nanotechnology in oral insulin.
Yeah, so it's a good question. First of all, you see, oral insulin, the major problem is whenever you are administering any protein through the oral route, there is a chance of degradation. If it is exposed by any way in the hydrolytic material like acids or even the enzymes, which will degrade the protein. So we all understand. Protecting it with these materials, it is quite possible. It is not impossible. But the important thing is the uptake from the intestine of this protected material, and then it should go to the blood pH and release is always difficult. Why I'm telling you difficult? Because you have to make a science behind it. You have to maintain a pH at which it will liberate or degrade. But same pH is also available in our intestine. Our intestines are very, very large, you know, or long, you can say, distance. It has a huge variation of pH, right from 1.2 to 8. So you understand almost all pH range is there inside the intestine. So if your target is, it will be in the blood pH, means it has to release at pH 7.2 to 7.4. So nearly neutral pH. But that pH is also available in intestine in some region, more particularly I can say jejunum region, even a part early ileum region, you will get this particular range of pH. So if it passes through duodenum and reaches in the area, your formulation will be lost. And if it can be absorbed before that and reaches into the blood, you know, that can release insulin. That can give insulin into the blood. But important issue is that there is a question of, you know, balancing, because when it is reaching in the duodenum, and you cannot guarantee it will also pass to the jejunum and ileum, and 100% it will be absorbed from the duodenal region at pH 6.8, because we are very nearing to 7. So there are lots of issues are there, but it is not impossible. It is possible to prepare, you know, insulin by oral route. But, you know, lots of investigations are required. Major problem is its absorption. Protection is possible because you can either lots of polymeric encoding, you can just protect it. But its absorption is very difficult, 100% from a particular region. And secondly, once it is absorbed, its release is also difficult, because you have to have a science behind it. You have to work with the pH, you have to work with certain methodologies, and that methodologies are very sensitive to all the region, including intestine to blood. So that's why it is not very successful, but people are trying.
Thank you, sir. And the next question is from the participant called Tejaswini. She asked that, sir, are these nanoparticles suitable to target brain mitochondria?
Yeah, it is possible. What is impossible? Because if you can bring it into nanoparticle, you are bringing it into the brain. So if you can bring it into the brain, and if it reaches into the cell, like as you have seen that it is reaching to the mitochondria. So it is quite possible. I don't think it is really impossible. But targeting, if you want, then you have to make it even a smaller size. And you have to keep it protected in the cytosol. That is, once it is reaching in the brain cell, it will remain protected, and then it will have a targeting molecule which will exactly bringing it to the mitochondria.
Sir, her question is that will it be suitable to target the brain mitochondria?
Yeah, it is suitable, it is possible, but there are two things. One is, I said that it should remain protected till reaching into the cytosol of the brain cells.
Right.
Second thing is that there should be a moiety, or there should be targeting molecules which will lead it to the mitochondria in the brain cells. So, because it is very specific about targeting mitochondria. So that means some some chemicals should be tagged to bring it into the mitochondria inside the brain cell. That I wanted to mean. And this is possible because there are chemicals available, and there are methods available by which you can target mitochondria.
Another question from the same participant is, sir, what are the biomedical applications which you have elaborated in your talk, sir? The other question is that does drug loading affect nanoparticles?
Of course, drug loading is very important. Like you are sending goods from one part to the other, and your boat has the capacity of 1 kilogram, and another boat is having the capacity of 40 kilograms. So obviously loading is important. Drug loading is obviously important for every formulation. And yes, this is obvious. This is obvious. This is important.
So The another participant called Ekta Daveshwar has asked that, she has asked that, sir, why are we comprehending L5 and L2 working successful after it targets the G2M phase? Like it not target the synthesis or G0, G1 phase and stop apoptosis?
Yeah, G0 phase is the very not common phase. It happens in particular type of cells. But G1 phase, see, well, normally we consider 4 phases: G1, G2, M2, and S. So G1, G2, MS. So what is important, that S phase, when cell is synthesizing these DNAs, more particularly for this preparation of the next proliferation, next division. Then G2 phase is not only this DNA, RNA, and proteins, and preparing absolutely for this cell division. And then M2 is the mitotic cell division, that is mitosis occurs. And then once cell has been divided, then another phase is your, that G1 phase, where the cell is just maturing, that is just cell growth, preparing, it's becoming adult. So you see, this 4 step of the cell cycle, stages of the cell cycle is important. So here you see, when your drug is working on the G2M phase, that means it is working in that division phase, mitotic division phase, your drug is working. And the paclitaxel actually is responsible for this arresting of mitotic phase. So that indicates that your work is leading to the right direction, because if you find that it is working in G1 phase, your drug is not supposed to work in G1 phase. It blocks the mitotic cell division. That means that there is some problem in your investigation. But here in this investigation, it suggests that it is blocking the mitotic cell division. That means it is working at the very right direction. That is important also. You have to understand that which phase of cell cycle your compound will work. That is important.
So she has one more question on the related to COVID vaccines. She asked that, are we using the same liposomal target therapy in COVID vaccines?
See, COVID vaccine, what is vaccine, you have to understand. Liposome is a carrier. But what is vaccine? Vaccine is basically you are introducing one antigen. That is vaccine. Because this antigen, once in your system, it will produce some antibody, but that antibody is required to destroy that antigen. But that is not enough. Their memory cells are there. The memory cells, they will remember that whenever this type of antigen will come, we will produce. Normally, what happens in COVID is when it's a new product. So new chemicals, when new antigen is coming, what happens? Body is not prepared for your antibody. So body needs time to prepare antibody. Body has to understand your antigen, whatever it is coming, foreign body. So it needs normally time, 3 to 4 days, sometimes for new cases. But in the 3 to 4 days, sometimes it aggravates certain kind of other problems, and that time other like cytokines and these things are trying to block it. Your innate immunity first try to protect yourself, and then cytokines. And that time what happens, huge cytokines releases, and similar type of, whatever the spike protein, similar sequence is also there in, our blood vessels, endothelial cells, also in liver, and some little bit in heart, and more in lungs. So the cytokines, whatever virus is damaging, virus is being killed. But the cytokines heavily damaging your system. Because they cannot understand which is virus, which is they understand this antigen, and they're heavily damaging your system. Lots of damage in the lungs, lots of your damage in the liver and other cells, and people, that because of the damage in lungs, people want oxygen. So if damage is less, you are giving oxygen. But if damage is more, you cannot, even given the oxygen, you cannot breathe it, you cannot take it inside, because your lungs is not that way, is able to take oxygen. So what happens that time? That time, ventilation, your oxygen is directly given into the blood. but simultaneously your lungs should be protected, because how long your oxygen will continue into the blood? Your lung has to be replaced, this is their job. Before replacing that job, if lungs collapse, you will die. So this is happening. So antigen is simply, it can be given by injection. You can give it by other means also. Introduction it into the body is enough, because there is a foreign protein. As soon as it is reaching, antibody will start production. But it takes for new case, it takes time.
So thank you, sir. And there are several other questions, sir. I'll be just few questions I'll be listing here. Dr. Rajesh Shastri, one of my colleagues, sir, he asked that which type of nanoformulation is suitable for treatment of blood cancer?
See, blood cancer is absolutely, you have to understand that blood cancer is a different thing. Blood cancer, you know, the major problem is the cell division, lymphoma or leukemia. The lots of blood cells, when it is generating, you cannot replace it, because it blocks the blood vessels and it can kill easily the people. Lots of, you know, what you call this, yes, hemorrhages takes place in different places. But you have to go to the source of it. That means your nanoformulation should go to the, you know, pit, or you can say to the places where from these cells are generated. For lymphocytes, or in case of leukemia, these blood cells, you have to stop this, you have to block this, like particularly, you know, spleen, particularly, you know, bone marrow. So there you have to bring the materials and you have to target those cells which are producing these cancerous cells. So it is little bit difficult, because to reach materials into the bone marrow is always difficult. There, you know, one option is there, that there are some what you call this chip type things, chemicals, you know, what is like silica or similar type of things, or porous chemicals, but you have to find out that in long term, they will, long run, it will destroy, it will degrade, and they contains the drug. And if you just bring it to the right location in the bone marrow, like implant, they will remain for longer time, release the drug, and locally they will try to remove the cancer cells. But this is not very, you know, this is complicated, because reaching into the bone marrow is another big job. It's not very simple, because you have to cross a big barrier again.
Yes. Sir, a few more questions, sir. The participant called Swapna Barai, she asked that when we talk about the nanoformulations, why do we find most of the application in treating cancer, and why not other diseases?
It is up to you. It
Yes,
is
sir.
up to you where you want to go. It doesn't mean that I am working with the cancer means everybody is working with the cancer.
Correct, sir.
This is thing. It is nano formulation you can use anywhere.
What
Even you forget about anywhere means in the body, outside the body also, in the tennis racket, even the swords, you can prepare by this nanotechnology. Nano formulation can be used anywhere. It can replace your what you call is this what is there in lens of the eye. It can do lots of work for you. So it is not that it is just simply for cancer. I am working with the cancer means it is not that anybody is not working in other field. Everybody is working in different fields with the nano.
One more question from the same participant, sir, but it looks very important. Can these nanosized targeted delivery system prevent the development of drug resistance? If yes, and how?
It is possible. Targeted drug resistance, you know, resistance, you have used a very big term. Very big term means you have to understand what is resistance. Resistance doesn't mean always it is like the microbial resistance, antibacterial resistance. Even your body cell becomes resistant to even different chemicals. Like I can say, even cancer cells get resistant with the cancerous drug. So you have to target the mechanism by which the cell is forming the resistance. So in case of microbial resistance, you can prepare the nano formulation like many, it can sometimes make a barrier it can just recognize that drug and destroy it. So by nanocarrier, if you just put it, they cannot recognize it. It will go inside the cell and it can kill the cells also. Or in case of cancerous cells also, the resistance formation is there because the EPR effect. So if you can target it properly, EPR effect, it can remain inside the cell, inside the cancerous cells, or inside the part where the cancerous cells are there. It can help to reduce the number of cancer cells quickly. So it is resistance of any kind of cells can be well tackled by this nano formulation. Well tackled.
Another participant called Tejaswini asked that what is the main cause of drug precipitation in nanoparticles?
Drug precipitation, it is not that always drug precipitates. Like paclitaxel, it precipitates heavily. Because you see, it depends on the drug solubility. Precipitation, why occurs? You understand that the simple physical chemistry, it is the solubility. So whenever you are trying to upload the drug in the formulation, you have to understand that you have to improve the solubility of the drug. If you enhance the solubility, it will help to upload the drug. Secondly, the quantity, if you take much more, obviously than the wanted, obviously it will precipitate. So you have to make a balance how much drug you will take, and then you have to improve the solubility. Obviously you can ward off this problem, you can remove this problem. It's not a big issue.
Sir, in your lecture, you have quite elaborately discussed about its the kinetics as well as the pharmacodynamics in both in vitro and in vivo model. One participant called Swapna Barais has asked that, sir, what are the safety parameters of nanoparticles to the embryos and fetus? So that's the safety aspect which needs to be addressed.
I could not follow this question, please.
So what are the safety parameters of nanoparticles to embryos and fetus? So what I understood is our question is on the safety of these nanoparticles, particularly on the embryos and fetus.
See, nanoparticles, you have to see that what purpose you are using. Fetus, this is they have barrier, they have placental barrier, so everything is not reaching to fetus, so that's why they remain safe. But you have to check also that your formulation, if you want to get the safety of the fetus, you have to check whether this is crossing the blood placental barrier or not. Most of the cases, it is difficult to cross blood placental barrier. So if it is crossing blood placental barrier, then you have to investigate that whether it will be harmful on the fetus or not. So first study you have to be sure that it is crossing the blood placental barrier. If it is not, there is no need for study. If it is, in that case, you have to also investigate the efficacy, effect of it on the fetus. This is important. Because I told you, nothing is safe in the world. Nothing is safe. No chemical is safe in the world, even water, even oxygen. If you take more oxygen, it will be toxic to your system. If you take more water, it will be toxic to your system. So nothing is safe for us. Obviously, with a balance, it is safe. Safety is just maintaining a balance between toxicity and minimum level of efficacy. So you have to maintain it, and you have to check whether it's safe. If you think that, okay, nanoformulation is not toxic to human being, it is not toxic to fish, it is not that. It could be toxic. You have to investigate it. I said that, like, in 2 steps you can investigate. First of all, it is crossing the blood-brain barrier or not. Yes or no? No, no need to study. Yes, you have to study on its effect on the fetus. First of all, in animal fetus, if it is not toxic, obviously you can go for the larger animal fetus, and then you can go for the humans.
Yes. Thank you, sir. There are some other questions which you already addressed, like that Rajesh asked that whether it can be possible for a neurodegenerative diseases like Alzheimer's, which you already mentioned that it is possible, it can be explored. Thank you for sparing your valuable time, sir, and giving an informative talk on this topic. Over to Manasadeepa.
Yes, sir. Thank you, Dr. Bannappa sir. I request Dr. Bannappa sir to hand over the certificate to our honorable speaker today. For this, I request to join Dr. Sharat Mukherjee sir and Parley sir to join for felicitating with the certificate to our honorable speaker today. Yes, please, sir. And thank you so much, sir, for accepting our invitation from APSE. And really, thank you so much for joining with us and sharing a wonderful and very informative for the budding scholars, those who are working in the nano-related work. Really, it's a very informative talk. And again, it's not to stress this word because his contribution is already, it has been registered in the scholar citations. It's more than 3,500. It's not so easy job to get such a number of citations in the Google page. So it gives you a lot of information. So if further any questions and queries, please feel free to write or you can approach to our speaker. It is available in the Google page. You can. Yes, Bannappa sir, please hand over the certificate to our speaker. Please unmute your audio, sir. Dr. Bannappa.
Thank you, madam.
Yeah.
Sir, thank you for your valuable contribution to this our society, sir. I request you to accept our certificate of appreciation, sir, for this delivering this lecture. Thank you very much, sir.
Thank you. I'm accepting it. Thank you very much. And I like to congratulate you all for organizing such type of things so that people can reach to the common people, those who are interested and working in the field.
Meanwhile, thank you, Dr. Bannappa sir and Vakade sir and Parley sir. Meanwhile, I request Professor Biswajit Mukherjee sir to select the best delegate by asking the best question you feel for today's event. By the time I request Dr. Vakade sir to, to give a few words about today's event.
Yeah, just before Vakade, I just want to say it is very difficult job for me to understand who is best in terms of delegates and who, in terms of questions also. But I think the most important question I received from the person who asked for this oral insulin formulation. This was a nice question.
Congratulations. Mr. Vinay, please find the participant who asked this question and please let us know. Meanwhile, I request Professor Vakade sir to give a few words about today's event.
Thank you, madam. Thank you, Mukherjee sir, once again for accepting our request to deliver a talk with our all APSE members and other faculty members. It is always blessings to listen to you in DIPSAR or any other scientific platforms, conferences. It is always nice to listen to you again. And this has given a very broad view of this nanotechnology and its applications. And it will encourage all the researchers, faculty members to again go into details of this research area, focused area, which is very important one. Once again, thanks all the organizing team and all my colleagues over here, Parley sir, and Joshi sir, Bannappa sir, Dr. Pawan sir. Thank you very much. Thank you, madam.
Thank you so much, sir. I request Professor Dr. Milind Parley sir to give a presidential remarks over the event. Parley sir.
Right. Thank you. It had been a nice session, very informative. And Dr. Biswajit Mukherjee, I heard after a long time, a very good stamina, long talk. So I'll not, add much. He has covered almost everything. And it's nice that delegates are participating. And we are continuing the tradition of giving the best delegate award who has asked the best question. So keep it up, APSE, well done. Right.
Thank you. Thank you so much, sir. After a long time, you have joined after his COVID, the sufferings. Thank you so much, sir, for your great presence over here. And Dr. Banappa, sir, can I request Dr. Professor Sharad Vakade, sir? Vakade, sir?
Yes, ma'am.
Yeah. So we never fail to give appreciate the participants. The roots of all goodness lie in the soil of appreciation for goodness. This is a great sayings of the Dalai Lama. With this, I request Professor Sharad Vakade sir to hand over the certificate to our best delegate of today's event is Dr. Jnanachandra. Yes.
Congratulations, Chandran sir.
Yes, a very short period we got. Dr. Jnanachandra, we are will send the certificate to you with your name. Yes, Professor Vakade sir, it's over to you, sir. Please.
Yeah, always actively involved in all these seminars, webinars. Thank you.
Chandran sir, if you are there, you can speak
Yes, you can
2
join.
words, sir. You can
Yes.
receive it. You can come, you can switch on your video.
He's available.
Are you there, sir? Chandran sir?
Yeah.
Okay.
Okay. Thank you. Thank you so much, sir. Feeling gratitude and not expressing it is like wrapping a present and not giving it. So without the end up or cannot be concluded. So for this, I request our founder secretary, Dr. Hanumanthachar Joshi, to give a vote of thanks to our audience. Dr. Joshi, over to you.
Yeah. So good morning to, good afternoon to everyone. It's already afternoon. And we are highly thankful to our esteemed guest today. That is Dr. Biswajit Mukherjee, a very well-known person in the field of the pharmaceutical sciences. Sir, thank you very much for your kind time and an excellent talk, sir. It was really a very, very beautiful one, and unfortunately, I missed a few beginning slides, but highly thankful to you, sir, for that. APSE is dedicated to bring the people who are really active in the scientific field. There are so many organizations in the country. They have their own the area of working, but we don't have any association, especially for the research scientists. We are very really fortunate to have you, sir, and once again thanks to you. And I also thank our Guruji, our the person who inspires us with both as well as research and as well as with the guidance, Professor Milind Parle, sir. Very nice to see you in such a nice condition, sir. Thanks to you for all the guidance and support. And on behalf of APSE, I also thank our dynamic vice president, Professor Sharad Vakade, sir, whose presence always boosts up our energy and as well as the stamina. Thank you, Vakade, sir. Nice to see you. And we have our one of the very important scientists, very young person, a person to be emulated as a model, especially for our younger research, the students, Dr. Bannappa, my very good friend, excellent, the Vatican's moderation, and as well as thanks for your time, Dr. Bannappa.
Thank you, sir.
And I need not thank, my own friends who are always involved in all our work, but as a formality, I should always thank Dr. Pawan Kumar, our brother, our very good friend, and as well as, a supporter. Thanks for that. And Dr. Varadarajan from Malaysia, Dr. Mehboob, who is also there. So we have people from different other countries also have joined here, and all the delegates. And once again, an excellent thanks to our friend and our leader, Dr. Mansadeep, who has been very instrumental in introducing again Dr. Biswajit Mukherjee and a lot of other speakers. And we have some more talks, to continue, which will be continued. At the same time, I request Dr. Mukherjee sir, kindly encourage our students, of our country, because the research, the culture is slowly, taking a sideline. It is decreasing. We request you to kindly guide our students. And thanks to everyone who have joined. And if I have not used your name, please excuse. And I can say Himanshu Joshi here from Uttarakhand, and all friends from all across the globe. Thank you very much, and have a nice time.
Thank you, Dr. Joshi. And as you mentioned, Precipitation, I think this is a wonderful platform, which I feel it because the two different gurus of the secretaries are of APSE are here. One is Dr. Parle sir of your guru. My mentor is Dr. Biswajit Mukherjee sir. So it is a really blessed day. I can say that that pioneer, which I have seen from past a decade, I can see his dedication and his work in the nano. It is enormous, and it is cannot be expressed by words. And of course, the people, those who are close monitor his dedications, can understand. Still, I have that the respect. And even you say, like, probably you might have been noticed that I'm very much open to talk or express my feelings very openly. But since I have that same respect and fear, I can say, scared to talk in front of sir. Sir, apologize if any interruption, if it has happened during the session. It's just because of the technical issue, because our technical person, very recently he got COVID. He recovered some few days back. And I want to thank Mr. Vinay, his enormous work, even after his post-COVID recovery. Just few days before only he got recovered, but he has his support is a lot meaning to our APSE, especially this event. I thank you so much, sir. Professor BM sir, we call it. Sir BM sir, thank you so much for accepting our invitation. And this have to be continued. And I pray the Almighty the same support and the strength which I give to my mentor. And we have to learn a lot from him and more to contribute our students. Thank you so much, sir, for being with us. And before I conclude this session, I would like to share this, the next event, upcoming event. Again, we never failed to give you the best always. Our next event will be on 9th July. Our speaker is Dr. Rakesh Kumar Sarma, Vice Chancellor of Savita University, Chennai. He's going to discuss a topic like 2-deoxy-D-glucose as promising candidate for tackling COVID-19. So we meet soon on 9th July. With this, I want to conclude and say thank you to everyone those who joined after your even having your tight schedule, giving your time for us. Thank you. Thank you so much.
Thank you. Thank you.
Thank you.
Thank you, BM sir, once again for your time and all the good support.
Okay, wish you all nice time. Enjoy your time. Bye-bye.
Thank you, sir.
Thank you.
Bye, sir.
Thank
Pakode, sir. Banapa, sir.
Thank you,
Parle,
sir. Thank you.
Yes.
Thank you.
Pawan,
Bye.
sir.
Have a nice day, sir. Bye.
Yeah, same here.
All the team members and delegates, please take care in this again COVID period.
Yeah.
Where I think
We can see Dr. Boosnuri also here. To all the people, thank you so much. And special thanks to once again Dr. Mansadeepa.
Oh, so it's a duty, not only research, nothing else.
Yes, correct.
Let me go
Yes,
with
sir.
this. Thank you.
Yes.
Thank you once again.
Thank
Yeah,
you.
bye. Bye. We'll leave it.